Good evening, everyone. My name is Alice Clayton, and I'd like to welcome you all to Menopause in the Brain. I'm the CEO of the McKnight Brain Research Foundation. We're a private philanthropy focused on research and education on cognitive aging. And I am delighted to be introducing Dr. Riley Bove, a practicing neurologist and clinician scientist at UCSF, and also a 2013 recipient of the American Brain Foundation Next Generation Research Grant. We're also joined here tonight by Dr. Maria Pia Campagna, a postdoc in neurology at UCSF and a member of Dr. Bove's lab, and one of two recipients of this year's McKnight funded research grants in cognitive aging and age-related memory loss. So I'm delighted to have everyone here tonight, and I just want to tell you a little bit about American Brain Foundation. It funds research across the whole brain, knowing that all conditions of the brain and nervous systems are interconnected. The foundation has a particular focus on funding researchers at the beginning of their careers, an early investment that continues to pay off year after year through the advancement of scientific knowledge. Webinars like this one tonight are opportunities to connect you with experts in various topics around the brain and neurologic care. I invite you to put your questions in the Q&A at any time, and after the presentations, we'll try to address as many questions as we can as part of the Q&A session. So let's move on to tonight's topic, menopause and the brain. We'll start with a presentation from Dr. Bove, providing some clinical and evolutionary context for the intersection of menopause and neurology. And then Dr. Campagna will present with her own research merging biology and data. So to tell you a little bit about Dr. Bove, she is a national and international leader in sex and gender aspects of neurology, publishing, collaborating, and lecturing widely on this topic. She's the PI of the first industry-sponsored phase 4 trial for a medication for use in pregnant and lactating people with MS, and led the first RCT in menopausal women with MS. She's founding director of the SAGE Neurology Program at UCSF, whose educational mission is supported by an NINDS R25 training grant. In the technology space, Dr. Bove has conducted innovative work designing, validating, and implementing digital tools to improve access to care, holistic measures of patient function, and as digital therapeutics. She's the co-PI on the Bridge Precision Medicine dashboards that deliver point-of-care personalized medicine. Dr. Bove sees patients at the UCSF Multiple Sclerosis and Neuroinflammation Center, where she provides comprehensive care for individuals with MS that is informed by their sex, their gender, reproductive goals, and more broadly by their life and their goals. She speaks French and Spanish. Dr. Bove, thank you so much for joining us. You have the floor.
Thank you. It's absolute pleasure to be here tonight to speak with all of you about one of my favorite topics, so menopause and the brain. This is really a topic whose sort of time has come, thankfully, and really looking forward to presenting some of sort of my perspectives on this topic, some of our studies on this topic, and also I think some of the really outstanding questions, and of course, passing some of the scientific baton on to Dr. Campagna to continue this work. So, real pleasure. So, here are my disclosures, and to just jump right in. So, menopause in the brain. Why are we so interested in this now? When I started my career and had my, fellowship, 15 years ago, there was not as much interest as today. And so, what has changed so that we're really finally paying attention to this topic in the way that it deserves, both clinically, societally, scientifically? So I think we're really starting to understand the importance of understanding menopause in the brain because we have an aging population. And so there's a lot of sort of, big, large scale studies thinking about this topic. There's also a surge in societal and scientific interest. I think really times have changed in this regard. And we have novel technologies that allow us to better understand how to do this work and how to understand mechanisms and such. And I think, just to sort of reflect on societal changes, we really now have women as customers, as patients, as scientists, policymakers, funders, clinicians, who all kind of know from lived experience that this is an important topic and that we need to put it to the forefront. And so, what are we talking about when we talk about menopause? I think it's just helpful to understand sort of these stages of reproductive aging. They were like super well described in this workshop 10, 15 years ago. And sort of this is the model that we use when we think about it. When a woman has her final menstrual period and then no periods for a year, we say she's postmenopausal, and the date of menopause was her final menstrual period. And that typically is 51 in Western societies. So about half of women have their final menstrual period before age 51 and half of them thereafter. And when we think about sort of understanding the clinical research and the science, we're thinking about a couple things. We're thinking about the age of final menstrual period. We're thinking about the type of menopause. So was it spontaneous menopause, or did it arise because the ovaries were removed during surgery or affected by radiation, or there were medications that caused early menopause? And then we also think about the age of puberty, and the time between puberty and menopause is the reproductive lifespan. And in general, what we're understanding is that the longer reproductive lifespan, the better for women's brains. Okay, so the news today is all around the menopausal transition, so perimenopause. But then we also want to think about post. And this is where estrogen levels are higher and then begin to sort of waver and then decline. And typically you measure someone being postmenopausal if you look at the follicle stimulating hormone, or FSH, which rises after the final menstrual period. We also then think about the postmenopausal lifespan, which is the rest of the lifespan. And so if you're an individual who has a neurologic condition before the final menstrual period, someone who has multiple sclerosis, epilepsy, migraines, myasthenia, then the questions are really going to be, what is my illness experience going to be during this perimenopausal transition? And then also, what is going to be my disease course after menopause when the estrogen levels decrease? What will happen to my disease course? Will it become more aggressive? Will the headaches go away, et cetera? And so just to give you some examples from our research in women with multiple sclerosis, we find that during, again, this perimenopausal period where the estrogen levels are wavering and then decreasing, we really see that this is a period of high clinical need for our patients. And they often ask these questions almost verbatim, almost in every perimenopausal, clinic visit. Is it my menopause or my MS or both? I'm just not sure. And what they're also asking is, who's going to handle my symptoms? People are worried about cognitive changes. Do I now have dementia as well as MS? And they're also just experiencing the subjective overwhelm. Something has to give. And so this is a time where people experience a lot of distress, even if their MS, their disease, is actually very well controlled. And if you just think about the overlap of all the symptoms of aging, all the symptoms of multiple sclerosis, all the symptoms of menopause, and then you add on like life on top of that, you can see there's really this region of overlap where people are experiencing a lot of symptomatic changes and really require management. So we'll go back to that in a moment, okay? One of the key features that people experience during perimenopause is almost this cognitive wobble. Things that used to feel intuitive, remembering names, staying on track, staying focused, seem to be shifting. And so a lot of times, historically, we've sort of just avoided this altogether. And a lot of times these symptoms really are really mostly perceived by the person themselves, even the person without neurologic condition. And often those symptoms, these cognitive changes, they fluctuate for a bit and then they stabilize. It doesn't necessarily mean there's going to be this loss and sort of a downward trajectory forever. But we do need to really take these symptoms kind of seriously and look at them carefully. And there are a lot of sort of clinical guidance and reassurance that can be given to women during this perimenopausal transition. And because it's not everybody requires cognitive assessments. Not everybody is going to develop dementia, even when it feels very distressing that there is this wobble. Okay, and this is a wonderful article that kind of talks about the counseling around this topic. Then when we think about the postmenopausal lifespan, so how will neurologic trajectories change? So in people who have established neurological conditions, this is where we see a little hint that loss of estrogen, loss of ovarian hormones can perhaps be represent one loss of resilience. Okay, so these are very, very subtle changes that we picked up really when we looked at biomarkers. So a blood-based biomarker, serum neurofilament light chain, and then a composite sort of examination. And we saw that after the final menstrual period, so this vertical line here and here, we saw that the slope of change just worsened a little bit. So the functional composite got a little worse. And the neurofilament light chain, which is a measure of nerve loss in a sense, got a little bit worse as well. This is not a major clinically relevant change for patients, but it does suggest that loss of hormones can represent one mechanism for loss of resilience. If we move to the conditions that really onset way after the final menstrual period and the conditions that we think about sort of in this long postmenopausal lifespan, okay, so women spend a third, a half of their life in postmenopause. This is when we're talking about dementias, mild cognitive impairment, stroke, Parkinson's, et cetera. And here the question is, will the time or the type of menopause affect risk of these conditions? And will menopausal hormone therapy help? Okay. And so this is a study that I did way back when I was a fellow. We were able to use this beautiful longitudinal cohort from Rush University, the ROSMAP cohorts. And we were able to look, so just to draw your attention to the figure here for a moment, at women who had spontaneous menopause, that's the red line, and then women who had their surgical menopause very early, under 38, between 38 and 43, 43, 49, or greater than 49 years old. And what you can see is that the women who had their ovaries out late, this is global cognition. So this is just a general cognitive measure of these women. When they entered the study, the women who had their ovaries out late had very similar cognition to the women with spontaneous menopause. Okay, the women who had their ovaries out early, their cognition was lower when they entered the study, and every year as they were followed over the course of the study, those differences became more and more significant. Okay, and what we were not the first to show this. The Mayo Clinic showed this and has shown this sort of really consistently and beautifully over the years. And many other groups have really shown the same effect, that early loss of estrogen or shorter reproductive lifespan from menarche to menopause, like I showed you, is associated with worsening in cognitive function, worsening brain volume loss, neuropathology, so sort of the markers under a microscope of Alzheimer's, and then higher risk of Alzheimer's, Parkinson's disease, stroke. And in the observational studies, we see that menopausal hormone therapy is protective. I'll go back to that in a moment. But essentially, we do see a strong signature that if the reproductive lifespan is abbreviated, there's an effect on brain health in the rest of the lifespan. Okay, so the reasons for this are just like really myriad. And what we see is if we look at early life exposures and then menopause and loss of hormones, that really in itself has a whole bunch of consequences. So increased risk of diabetes or higher body mass index, changes in the bones, muscles, changes in blood vessels, changes in heart function. All of those things are relevant to brain aging. Okay, they're relevant to brain health. But the question that we also have as neuroscientists, and this is where we're really moving into the mechanisms, is does the loss of the hormones itself affect disease-specific neuropathology or general brain wellness in a way that affects brain health in later life? Okay, so this is just to show sort of the complexity of the analyses that we have to do to try to disentangle potentially more direct effects on brain versus more indirect effects. So the mechanisms are just beginning to be uncovered, and stay tuned for Dr. Campagna's talk, thinking about how menopause shifts lifespan risk factors. What are the epigenomic changes? How does the way that genes are sort of expressed, how is that affected by menopause? What is happening really in the synapses or the sort of neurons? And what factors may provide some resilience, okay? And so if we talk about resilience, we have to think about the fact that menopause is actually an evolutionary superpower. Okay. It's not just sort of like a loss of, sort of reproductive failure. Okay. This is actually a evolutionary adaptation. So we have women who stop childbearing and then who live many, many, many years beyond that. And the reason for that is sort of a version of what's called the grandmother's hypothesis. So when women stop childbearing directly and live for a long time, they can invest their wisdom, their knowledge, their resources, their strategies into raising the very, very dependent babies, their children, grandchildren, all the way to adulthood. Okay, so shifting from directly childbearing to raising their offspring and their grand offspring to adulthood. Okay, and so this is really sort of a very kind of novel concept. Very few species have such a protracted long period of postmenopausal lifespan. And so it suggests that if estrogen, loss of estrogen were just so bad across the board, then how would we have these brains that really are designed to fulfill very sort of important cognitive roles postmenopausally? Okay, there must be resilience factors as well, and that's something that we're really interested in. So the question I get all the time, menopausal hormone therapy, good or bad? And so as perhaps people know, when we really look at the trials, when we really look at systemic, meaning in the whole body, menopausal hormone therapy, when given to women under 60, within 10 years of menopause, so we're talking about estrogen, and the preferred mechanism is transdermal estrogen. And then if women still have a uterus, give progestogens as well. That is really associated with a lot of improvements in RCTs, so randomized clinical trials, best quality evidence, both during perimenopause and for general health and the rest of the lifespan. Effects on brain. In the observational studies where we just follow cohorts, we see a protective effect. In the interventional trials, we don't see a protective effect, but that may be because we haven't followed patients long enough. Okay. And so Dr. Campagna will pick up some of those themes as well. So finally, just in the clinic, what do we do? We have, evidence-based management for well child baby visits, for well baby visits, for well child visits, for pregnancy visits. How about having some evidence-based comprehensive management for well woman visits at midlife? Okay. And so from a clinical standpoint, I really start, guiding patients around age 45 that something may be on the horizon, and not to worry, we'll handle it together. Really comprehensive symptom management. And this is something that it really is in the neurologist's lane to do, because these are all things that are affected by neurological conditions and that affect neurological brain health. So sleep, hot flashes, mood, fatigue, cognition, bladder health, etc. And then the Lancet came out with these 4 modifiable risk factors for late life cognitive impairment, late life brain health. They forgot sex, gender as part of these risk factors. And so let's sort of look at these wonderful modifiable risk factors, add in, sort of appropriate management of women throughout their lifespan. Think about these compartments that only affect females, breast health, cervical health, et cetera, to have a really comprehensive management. Okay. And then, so we talked about why now. So what do we do now? So we really need to drill down on understanding the clinical impact of menopause within and across diseases, educating our clinical workforce. So that's something that's really a priority for me, is making sure that everybody who takes care of females of menopausal age and postmenopause understands what these factors are. Implementing new approaches in the health system. We need to maintain the funding on this kind of research to promote sort of, healthy women, healthy grandmothers, okay, healthy families, empowering our patients, and then building the cohorts to really ask the important scientific questions and addressing those mechanisms. So with that, I'll stop and I think hand the baton back to Dr. Clayton.
Thank you so much. That was so interesting and a wonderful talk. I guess I would ask, what would for people in the audience who are going through this transition, or think, are thinking about it in the coming years, what would your best advice be in terms of managing their own health and also their sort of expectations, and how to sort of advocate for themselves?
Yeah, I think we're really seeing a growing workforce who's interested and, who will partner with patients. So I think finding the right clinicians who recognize the importance of this is really key to brain health over the rest of the lifespan. And then really addressing symptoms sort of comprehensively one by one. I think social isolation, social wellness, lifestyle, exercise, nutrition, sleep. If people are not sleeping well, and we know that after menopause, women have higher risk of sleep apnea, restless legs, bladder plays a role, mood plays a role. Sleep is just so central to brain health and wellness. So if the hot flashes are interrupting sleep, they need to be managed. If sleep is not, there's cognitive behavioral therapy, CBT, insomnia interventions, if people don't want to take medications. So there's a number of sort of lifestyle preventive approaches to managing these symptoms to promote brain health.
Wonderful. Thank you so much for that advice. And I'm seeing a few questions in our Q&A. Keep them coming, and just go ahead and keep populating that Q&A, and we will take the time now to move on to Dr. Campagna and then take all of those questions at the end. So Dr. Maria Pia Campagna, PhD, is a postdoctoral scholar in neurology at the UCSF School of Medicine. She completed her PhD in translational research at Monash University in Australia in 2023, earning the Vice Chancellor's commendation for thesis excellence. Her doctoral research used bioinformatic techniques to identify genetic and epigenetic factors influencing clinical outcomes in multiple sclerosis. In 2024, she joined UCSF to investigate multi-omic mediators of postmenopausal neurologic outcomes under the mentorship of Dr. Bove and Dr. Catherine Rankin. Her current research is funded by the McKnight Brain Research Foundation through the American Brain Foundation. I give it over to you, Dr. Campagna.
Thank you. Okay, so I'm going to shift a bit away from the clinical side that Dr. Bove just took us through expertly and start to think about the research that we're doing that has been funded by the McKnight Brain Research Foundation and also the American Brain Foundation, and start to think about how we move from risk factors to mechanism and underlying biology. And so I think what's important here is that I'm going to go through risk factors, talk a bit about causality. So, do these risk factors actually lead to the brain health outcomes that we're observing postmenopausally? And then, how can we harness that using different scientific and multi-omic techniques to really understand what we can target therapeutically? And I think why this is important is because, just like Dr. Bove touched on with the Midlife Lancet kind of modifiable risk factors, menopause is not something that we can choose to not have. And so really understanding risk factors in a granular manner is important to understand and to inform the biology underneath them if we're going to intervene in women's midlife health. And so when we think about menopause, I think we also need to think about not just one uniform biological event. And so when we want to think about biology, we need to actually separate out the type of menopause. So thinking about spontaneous versus surgical subtypes and the indication for those subtypes, as well as hormones other than estrogen. So things like progesterone and also testosterone. Reproductive lifespan in terms of the number of years between puberty and also menopause, and also understanding if it's either end of that spectrum, which is of interest mechanistically. We also, I think, have a long way to go in terms of looking at symptoms and how that relates to late life brain health, and then also kind of detangling the impact of therapies, so hormonal, non-hormonal, so things that treat menopause symptoms, and also anti-estrogen therapies that I guess induce this hypoestrogenic state, which is similar to menopause, and harnessing that to understand how these risk factors inform our brain health in later life. And so when we think about studying these risk factors, what makes it really hard is that the lag time between menopause and late life cognitive decline is often several decades. And so this is then confounded by different comorbidities, different confounders, as we saw, as Dr. Bove showed, with the many kind of, inputs and outputs of reproductive aging. And so if we want to actually understand in granular detail what these risk factors are doing for our late life cognitive aging, we can actually basically use surgical menopause and these anti-estrogen therapies as, I guess, these naturalistic quasi experiments to strengthen strengthen the evidence of causality. And so we don't just want to know that these risk factors of early surgical menopause are associated with late life dementia, but we also want to understand if that's actually the cause. And so once we understand that, we then can move from causality to mechanism. And so that's really what I'm interested in the multi-omic work that I'm doing. And so as neuroscientists, when we're trying to understand underlying mechanism, we would usually turn to rodent models. I think this is particularly limited in the context of menopause because our rodent models often actually, I guess, mirror premature ovarian failure if menopause is chemically induced, or we can also see a more similar model of surgical menopause if the ovaries are surgically removed from these animals. We also know that rodents, particularly mice, only have an estrous cycle. And so if we're interested in the interplay of different I guess, ovarian hormones beyond estrogen. That's something that we can't study in these animal models. And also, as Dr. Bove touched on, the human postmenopausal lifespan is incredibly unique evolutionarily. And so this is something that we can't study in these animals that don't actually have that protracted postmenopausal lifespan. And so it means that we need to study humans and study a target tissue of the brain to directly understand the mechanisms linking menopause to cognitive aging. And so this is something that we've thought about a lot, not only in our lab, but also at UCSF. So about this time last year, we convened a range of experts in different disciplines from endocrinology to bioinformatics and in cognition. And through the UCSF Sex and Gender Enriched Neurology program, which is directed by Dr. Bove, we actually laid out a roadmap of what we thought was important to move from causality to mechanism for women's for the link between menopause and late life brain health outcomes. And so the first direction is a critical pillar where actually what we identified was an infrastructure problem that proves to be a big barrier to this work, and is actually just identifying data sets that we can use to study this problem. So in neuroscience, a lot of the time, if we want to understand mechanism further to animal models, we can use brain banks. And so that's kind of a brain that's collected at autopsy, post-mortem. And this is done a lot for dementia research, for Alzheimer's research, and that allows us to do some deep multi-omic profiling on those tissues. Unfortunately, when these aging cohorts were designed and they were building these brain banks, what wasn't a priority was reproductive aging. And so what we found as we delved into these brain bank cohorts is that they actually didn't have sufficient data on menopause for us to be able to study the mechanism that we were after. And so on the other side, when we think about cohorts like the Women's Health Study and the Women's Brain Health Initiative that are really focused around, I guess, women's health, particularly in midlife and throughout the menopause transition, what we found is that the brains weren't collected post-mortem, and that means that we actually weren't able to study mechanism in the brain tissue, the target site that we're after. And so this ROSMAP cohort that Dr. Bove mentioned briefly that she found her surgical menopause and cognition effects in is actually incredibly unique because it's the only cohort that not only gives us cognitive information while the women were still alive, but also reproductive information from before they were enrolled in the study. And then we also have brain tissue that was collected after death. And then that was then profiled multi-omically, and we can then link that to our reproductive outcomes of interest. And so this ROSMAP cohort is really integral to the second research direction that we identified in terms of being critically important to understand the mechanism of menopause and its impact on late life brain health. And so here I'm really going to talk about the project that was funded by the McKnight Brain Research Foundation, which is mapping the molecular signatures of menopause on the brain through integrated multi-omic profiling. And basically what we're trying to do here is link reproductive lifespan. So not only the age of menopause, but the number of years that a woman is reproductive, so between puberty and menopause. Link that with the data that's available in our brain tissue. So different genetic layers and epigenetic layers from the brain tissue after death. and then also link that back to cognition in late life. And so, what's really unique about this study is that we're triangulating these three factors, and that allows us to move from reproductive lifespan and age at menopause being associated with cognitive outcomes in late life to actually understanding what the molecular state of the brain was at the time for different cognitive aging profiles. And so, just to dive a bit deeper into the ROSMAP cohort, so this is run by Dr. David Bennett at the Rush University in Illinois and Chicago. And basically, they were recruiting either nuns or women in retirement homes across Illinois who had no known cognitive impairment at enrollment. So these were cognitively healthy women at enrollment, and throughout the study, they either developed dementia or they did not by the time of death. And they were tested annually with a battery of 21 cognitive tests. They took routine demographic and lifestyle surveys and also provided the reproductive history and information on menopause hormone therapy. And then they had over 90% brain donation at death, which means that we were able to do this really in-depth multi-omic profiling. And we had about 204 women for which we had all of this data available, as well as their reproductive history. And so what we're doing here is not only looking at bulk brain tissue, so we're looking at the prefrontal cortex specifically, which is the region of the brain really involved in higher order thinking and cognition, but we're also looking at the specific cells within the brain. And so we don't just want a kind of whole brain signal, but we want to understand what is happening in each individual or different cell of the brain, which will inform future therapeutic targeting. And so we're then going to link these molecular changes in these different cell types to our cognitive slopes over time. And so we're going to link this to global cognitive decline in line with earlier work that Dr. Bove spoke to, but then also link that to domain-specific cognitive slopes, so different domains of cognition and memory as well. And once we do this, we have a molecular picture of this relationship between the reproductive lifespan and cognitive outcomes, and whether this is mediated by different molecular profiles. The questions that we can then probe include the effect of surgical versus spontaneous menopause, and also the effect of hormone therapy. So basically, do we expect hormone therapy to modify the molecular signature that we found to be associated with, for example, accelerated cognitive aging? And so our core analytic sample of 204 women spans both spontaneous and surgical menopause. So only about 30% of our cohort experienced surgical menopause. And if we just look at their age as menopause on the x-axis here, basically the takeaway is that our median age at menopause for both of our women, for both of our groups of women, are as we would expect. It's about 51 years for our spontaneous menopause group, and that's slightly shifted to a younger median age for our surgical menopause group. But basically, we can see that there's a wide variability in age at menopause, and that's what's going to be really important for us to probe the different associations and mediation with molecular profiles and cognitive trajectories. And then basically after menopause, there was a period of a number of years before they were enrolled in the ROSMAP study. And then after enrollment, we're then able to track their cognition and whether they were diagnosed and at what age they were diagnosed with dementia, to then tie that back to age at menopause and the molecular profiles that we've observed. And so in terms of preliminary results and what we're seeing with biology, pathways that we see increased in earlier menopause include excitatory synaptic signaling and glutamatergic transmission. So I would say a neuron-based signature in earlier menopause, and then pathways that we're seeing in later menopause include extracellular matrix remodeling and adhesion biology, immune signaling, but not necessarily a pro-inflammatory signature, but also mitochondrial OXPHOS signaling as well. And so these are very kind of preliminary results, but biologically make sense. And what actually is most striking is that we're not just seeing kind of this, I guess, upregulation or downregulation of the same pathways in earlier or later menopause, but actually completely different biological signatures at this stage, which we will then be able to relate to different cognitive aging profiles. And so, in terms of the clinical impact of this work, what we hope this could inform by looking at our different menopause subgroups is firstly, menopause hormone therapy guidance after surgical menopause. So at the moment, menopause hormone therapy after surgical menopause is recommended up to the age of 50. It's not personalized. And so what we hope is that we could actually characterize how menopause shapes gene regulation in different cognitive aging profiles and build a molecular profile of this. And then, like, subsequently, we could identify women who have particular molecular risk profiles and see if they perhaps need a hormone therapy beyond 50 years old or adjunct therapy targeting the vulnerable pathways that we've identified. And then in terms of future works and scientific kind of areas of interest, we could then compare healthy and accelerated cognitive ages and then see if basically epigenetic changes have good therapeutic targets. So we could use these epigenetic changes as biomarkers to guide treatment, and there's precedence for this with cancer treatment in the brain, so glioblastoma, but also guide non-hormonal interventions that might target the pathways that we've identified. And so, in terms of kind of linking risk factors, causality, and underlying biology, what we're actually starting to find is that the risk factors are going to inform what we pay attention to in terms of causality. And then this causality is then informing our underlying biology, which leads to the interventions and stratification that can be then applied to different patient populations based on these original risk factors as well. And I think what's also very important to highlight is that these aren't going to be, I guess, molecular profiles of menopause, but they will be molecular profiles of cognitive aging. And so that means that they're going to be applicable to, a broad population of people, including with further study, different populations, including men. And so the work we're doing now not only, I think, will inform, women's brain aging, but also everyone across the board will hopefully benefit down the line. Thank you.
Thank you so much. That was wonderful. Really appreciate it. Just a quick follow-up question. You talk about the criticality of, when that menopausal age is, whether it's spontaneous or early. Is your sense that it's more of a acceleration, deceleration on the same trajectory, or do you think it's really sort of depending on when it happens, a sort of an alternate trajectory? I mean, that other sort of biological pathways are triggered. So it's more than just a quantitative, I guess, shift in the phase, but a real qualitative as well.
Yeah, I think that's a really good question. I think my intuition probably leans towards the latter, but I think there's also a separation in terms of the underlying biology and actually what we're observing functionally in terms of cognition. I think the shift in terms of cognition is probably a bit of a dose response, if you will, a bit of that shift, as you mentioned. But I think the underlying biology is probably different, and that's what we're starting to observe with this study as well. So I think it's probably detangling kind of the cognition and the overall function from the underlying biology. I think that will kind of be separate.
Yeah, fascinating. That's such a critical question, has so many clinical implications for folks. I wanted to get to the questions that are in the open in the Q&A. Maybe I'll start one that's a little bit, some of the ones that are like a little bit closer to the data questions first and kind of move backwards. Kelly asks, have we studied women more than 10 years post-menopausal to know whether HRT has benefits?
Great question. So the Women's Health Initiative, right, started HRT or MHT these days, same thing, in women in their 50s and also women in their 60s. And what was found is that when it was started in women in their 60s, whose estrogen receptors really hadn't seen estrogen in quite a while, those women had a higher risk of clots, so strokes, blood clots, and also elevated risk of sort of like, brain aging. Okay, so then what happened is MHT, HRT was stopped for everybody after the Women's Health Initiative memory study, the WIMS. So what is now kind of, we're tiptoeing back, and actually the last few years we're floodgating back to giving menopausal hormone therapy to women within 5-10 years of final menstrual period who are under 60. So what do we know about those who might start menopausal hormone therapy later. I think there's a difference if you're starting it versus if you're continuing it. So if you have been using it in your 50s and the whole time, and then are thinking about continuing it, we are thinking about increased risk of clotting with aging, increased risk of heart disease with aging. And so thinking about an individualized decision around whether to continue the menopausal hormone therapy. In women who haven't been on it at all and who are now considering starting, I think to date, the evidence is not, doesn't really support starting that in the 60s. Just because it might be a bit of a shock to the receptors. And so in terms of achieving the benefits that you want, in terms of sleep, in terms of bone density, in terms of those things, those might be better pursued using non-hormonal treatments. But for people who are continuing them, we're really starting to see the field shift to where it's like, hey, if you've been on it for a little while and you've been doing really well and you're in a low risk category, it may be okay to continue. And I'm definitely seeing those trends in clinical practice.
Great, thank you. And so I think it partially answers one of the questions that says, have there been any benefits to brain health seen for starting HRT in women greater than 60? And based on your answer right now, no.
Why
Um,
not?
mhm.
Yeah.
Okay. There's another question here. It says, are you able to speak to which estrogen type has greatest effect on neurotransmitter profile changes in early versus late, more immune in menopause, i.e., E2, E3 versus GPR1, GPR30 effect? Does that?
That's a big question.
Yes,
That's a
it is
great
a big question.
question. So let's talk about the types of estrogens for a second. The bottom line is no, not really. Don't have a great answer for you. But let's walk through it a little bit. So when we talk about estrogens, we're actually talking about a couple estrogens. Okay. So we're talking about estrone, which is one, estradiol, which is two, and estriol, which is three. Okay, talking about the composition of the estrogen, how many rings they have, one, two, three. And estrone is continues to be produced after menopause by fat cells. Okay, so estrone is one form of estrogen that continues to be produced after the ovaries no longer produce estrogen. Estradiol is sort of the more sort of the one that has been the best studied in terms of its effects on brain. Estrone in general has not been as well studied. And then during pregnancy, estradiol rises, and so does estriol, third. And so that has also been studied in selected populations in mice and in women with MS specifically. So what we're typically talking about is estradiol. And then we're talking about estrogen receptors. So ESR, the receptors that receive the estrogen are either ESR1 or ESR2, otherwise known as estrogen receptor alpha or beta. And like in mice, there's different findings regarding ER alpha versus ER beta. In humans, it's just a little bit harder to see because we don't have the best, live imaging of estrogen receptors in the brain. There are some PET studies of estrogen receptors, but the PET ligand, the tracer, the way to look at estrogen receptors is not very good. So we don't really have a strong answer regarding some of these things. There are definitely some drugs that have been developed that target more estrogen receptor beta with an idea that that might be more neuroprotective, et cetera. But really clenching it is a little bit more challenging. I don't know if Dr. Campagna, you want to add anything from the sort of multi-omics bioinformatics side?
Yeah, I think I will just say it seems just in terms of the immune aspect of the question, in terms of early or late profile changes, it seems that the G protein coupled estrogen receptor probably has more of a role in that relative to ER alpha or ER beta. But I think that's something we can definitely probe with the study and the data that we have. We're definitely looking at all of those receptors and the genes that underlie those proteins. So definitely a question to note down that we can look further into.
Great, thank you. So there's a couple of interesting questions around this concept of brain fog. So I'll kind of put them together. So could you describe, what is meant by when people commonly use that term, and how does it manifest during perimenopause, menopause? And then importantly, how do I know that that's sort of normal brain fog, and when should I be concerned?
Yeah. So I think um, that let's address the concern part first. So the things that would be more typical would be having tip of the tongue, like I just can't find the word, word finding, um, finding oneself, having a harder time staying on task, kind of jumping around. Um, a little bit of the going into the room and wait, what was I doing in here again? And a lot of people talk about unmasking of ADHD at menopause. So they talk about just sort of like their ability to sustain attention, to focus, just it is less. So those are all things that are common sort of cognitive concerns. Something that would be more worrisome would mean like a clear change in language, or really sort of, getting lost, or things that would really be sort of like a big difference. So often, people, when they talk about sort of the cognitive concerns, I mentioned the other one that's very common is overwhelm. Like, if you're sitting there focusing on something, you can do it pretty well, but other people come into the room or add other stressors, and it's very easy to become overwhelmed, especially if fatigued, et cetera. Those are all things that we hear very, very commonly during perimenopause. So those are the cognitive concerns. And so, those would be reassuring. The ones that would be less reassuring would be sort of frankly like getting lost, or really having a hard time creating a sentence, or things like that, or having relatives really worry. Okay. A lot of the cognitive concerns that happen during perimenopause, loved ones may notice them a little bit, but it's really a lot on the person who's experiencing them. And so then those are the cognitive concerns. And then people also just talk about fog, brain fog, cognitive fog, things like that. So that's a couple things. It's feeling cognitive fatigue. So just like you're just more fatigued, like just thinking is more laborious. It's also like comes from just like general fatigue, where people are not getting good sleep. They're up anxious, or having to go to the bathroom, or hot flashing, etc., and just not getting good sleep. So there's a component of fatigue, there's a component of cognitive fatigue, and there's a sensation of just not being quite sharp and present. So the fog is sort of a foggy concept in a way, and it incorporates like inefficient cognitive processing, fatigability, and just not being quite sharp. Mm-hmm.
Thank you. There was a couple of interesting questions here, mostly about co-occurring conditions and your advice in terms of HRT. Are there any possible benefits for
Mm-hmm.
HRT in patients with existing neurodegenerative diseases such as
Mm-hmm.
Parkinson's or Huntington's?
Yeah, it's a great question. So, in general, sort of Parkinson's onset is kind of, more in the postmenopausal lifespan, right? So 60s, 50s, 70s. By that point, fewer people are on menopausal hormone therapy. And so we don't really have a good sense of how that might change things. Parkinson's is one of the conditions where we see in the observational cohorts that people who have, early surgical menopause, they have a higher risk of Parkinson's, and menopausal hormone therapy in those cohorts seems to have some benefit. So it does seem to have sort of like be part of this sort of protective story when within the window of opportunity, et cetera. Once the person has the condition, it's not so clear what the hormone therapy would do on a brain specific way. Like if it affected other things, right? Again, sleep, bone health, heart health, et cetera, that could play a role back into wellness. But in a Parkinson specific way, we don't really have, any data that it would be helpful. Mm-hmm.
Thank you. There are a couple questions in here that are sort of cohort related, and I think the first one actually says, Riley, can you comment on the N dichotomy in the WIMS trial and the observational trials on HRT? And there was another one, I think, about sort of race, ethnicity,
Mhm.
diversity in another one. Maybe I
Mhm.
should take just the opportunity if you could answer that specific question, but also sort of comment about the use of and the both sort of um, different strengths that come from different types of cohorts and how those cohorts contribute to science uniquely for both your research and your colleagues.
So it's interesting. So, um, in terms of cohorts, there we really need to ask these questions. We need these really large, comprehensive cohorts. As Dr. Campagna was describing, we really don't have very many. And so we need to use the ones that we have and then understand what the limitations of those cohorts are so that we can kind of like, well, this cohort is great for this, but we need to adjust for that, right? And this cohort has more of this information, but it's missing these years, right? So we kind of have to triangulate and try to get to a place where we're looking at the same sort of lines of, mechanism across the cohorts. So I think that's sort of a challenge and sort of like organizing and collaborating that we have to do. Um, with respect to the question around the dichotomy, so again, so the Women's Health Initiative memory study again was the Women's Health Initiative, where they really were giving, they were doing a trial, an RCT controlled trial of menopausal hormone therapy versus placebo versus the observational studies. You're just kind of looking at these existing cohorts, the sort of long-term followed ones, and just seeing. The problem with the observational studies is um, if you're someone who is sort of has better access to healthcare, who has better access to evidence-based research, who feels well enough to go pursue those questions, then you're going to be more likely to be on menopausal hormone therapy. And so your wellness is going to affect your choice of taking this medication. And so if you look at the study, the fact that you're well and you're taking the medication could be flipped causality, right? So that's a problem with observational studies. And the interventional trial, the Women's Health Initiative, again, that was really randomized and controlled, but it was done kind of in the wrong population. There is one other interventional trial, the KEEPS trial. So the KEEPS trial is sort of a more modern trial where they gave systemic menopausal hormone therapy to women sort of, much more in line with practice today within 5-10 years of their menopause, et cetera. And in that trial, it did show beneficial effects on a number of things. It didn't show beneficial effects on cognition, but they're only, 5, 7, 8, 10 years into the follow-up period. And we really need, like, as was said before, like 20 years to really see if it makes a difference. Mm-hmm.
I think I might also just comment on the different race and ethnicities question, which I just answered in the chat. I think that's why I disappeared. Our ROSMAP cohort is largely white non-Hispanic, and that was kind of a targeted recruitment strategy because they wanted to look at genetics. And genetics is confounded by different ancestries. And so since then, there has been an extension study to targetedly recruit non-white populations, but it's just that we don't have as much postmodern brain data for those populations. And so that's why for this kind of deep molecular phenotyping, we are still using the core ROSMAP cohort that's predominantly white. But it is something that we obviously need to address moving forward and in our future investigations too.
Mm-hmm. And just to add to that also, if we think about perimenopause, there are really very many different cultural factors that influence the experience of perimenopause. So when I was an undergraduate studying anthropology, this really landmark study was done by this medical anthropologist in Canada, Margaret Locke, and she looked at the experience of perimenopause in Japanese women compared to Canadian women and US women. and found that the symptoms, the hot flashes, the symptoms themselves were different, the frequency and severity of them. And we see that in US in different, in people of different ancestries and sort of different, uh, cultures, food intake, nutrition, all these factors are reflected in the actual physical symptoms of perimenopause. So there's a huge sort of like social, environmental, uh, trace there that also is very interesting when we think about sort of like these different contributors to brain aging. Mm-hmm.
That's really fascinating. Is it thought that I mean, it's sort of that the psychosocial influencers are sort of moderating the physiological effects, or it's not clear?
Different things. So, sort of social stress, smoking, habitus, specific nutritional, nutrient intake, sort of diets throughout the lifespan, exposure to, soys and, phytoestrogens, like many different factors that are related to sort of like culture and where one grows up over the lifespan and how that might affect these things. And then also number of children and, exposure to OCPs and all these things. Yeah. Uh,
Yeah.
Many different social cultural factors.
Yep. There's some interesting questions here, in addition about sort of the cognition one, and I thought maybe I'd frame it this time more as some of those modifiable risk factors that you had mentioned earlier. So there's a question here about cognitive reserve, and does it play a role in the development of brain fog and MS? And if you could answer that, and also maybe just talk about it more broadly in terms of, are there modifiable risk factors? Is there an interplay there with dementia and the reproductive age? I guess that's an open question to a certain degree, but if you could comment on what we know about modifiable risk factors as um, a mediating
Yeah,
factor.
yeah. So my hope is really that we think of perimenopause as a modifiable risk factor in the sense like perimenopause management, in the sense that if you have someone who's going through midlife and they have changes in their mood, changes in their sleep, that's going to translate into changes in their exercise, changes in their nutrition, that's going to like it's going to add up to a lot of different changes in their health status. And so my hope is that perhaps for some people through, hormone treatment, but also for others through like reducing that symptomatic exacerbation and like, really doubling down on the preventable, modifiable changes that we can get people on a better trajectory for cognitive aging. So because there, people experience a few of these symptoms at midlife, and together it can really throw their trajectories off. And so the idea is, can we peel people back onto a healthier trajectory if we're managing the hot flashes? You're not having someone whose sleep is interrupted, for a year, right? Like, and then, downstream patterns. So that's the hope, that we think about how we pull, rein in all those factors at midlife into more comprehensive management.
I know we're coming up on the hour, but I thought, I'd close out by asking you all to comment. This is with Dr. Campagna as a early career scientist, and both of you sort of studying such a critical topic here. Where do you see the potential for science on this topic over the next 5 years, and how you know, folks can sort of help to advance the science, even if they are not doing the research themselves?
That's a great question. For me, I think a lot of it kind of lies in what we're able to study in terms of the data that we're able to get, and obviously that in turn comes with the cohorts that are available. And I think that's not only with postmortem molecular data, but also how we can link that to, I guess, correlates during the menopause transition, whether that's in blood instead of the brain, and then start to link that to a range of different data points and outcomes at midlife as well. So. I think for me, I say the next 5 years, a lot of it is a push around what's the ideal kind of like data set and data collection that we want to build, and kind of like seeking funding to do that. Um, yeah.
Great.
And I just, I think there are 1,000 things to be done. There's 1,000 ways that we can collect data. We certainly need the really rich, comprehensive approaches, but there's many other approaches as well. The real challenge has just been that people weren't looking. Once you start looking, the data are everywhere. The questions are many, and there's a lot we can do. So it's just really encouraging to see so much enthusiasm finally for these topics.
Wonderful. Thank you so much. Well, I apologize to the audience members where we were not able to get your questions. There's clearly a lot of interest on this topic, and I want to thank Dr. Bove and Dr. Campagna for an amazing, two talks and being so generous with their time. I'd also like to thank the American Brain Foundation for organizing this, and to encourage you all to follow them on social media to stay up to date on brain disease information and research. Thank you all so much.